Have you ever just stopped and wondered what it would take to actually change the fundamental code of life? I'm not talking about science fiction. I mean in a real-world lab. Well, today we are diving deep into the incredible science of gene transfer. The actual techniques scientists use to move DNA from one organism to another, literally rewriting the instructions for life itself.
So here's the game plan. We'll start with the essential toolkit for cutting and pasting DNA. Then, we're going to see how scientists get clever by borrowing from nature, using some brute force, and even a little chemical persuasion to get genes into cells. We'll wrap it all up by comparing these strategies and looking at what this all means for the future.
It really all boils down to cracking one single problem. How in the world do you get a new piece of DNA, what's called a transgene, into a living cell and make it stick? That's the challenge, and that's exactly what we're going to unpack today.
Okay, so before we can even dream about moving a gene, we have to get it ready for its big trip. You can't just reach in and grab a strand of DNA. Nope. You need a highly specialized set of molecular tools to carefully cut it out and prepare it for its new home.
Think about it like this. Imagine a gene is one critical sentence in a massive 1,000-page book. To move that sentence to a different book, you'd first need some way to cut it out perfectly, right? And then you'd need some glue to paste it into the new page. Well, in genetics, we do the exact same thing, but with special enzymes.
And this is where the genius of it all really shines through. We basically use two types of enzymes. First up, we have restriction enzymes. These are like hyper-precise molecular scissors that recognize and snip DNA at very specific locations to isolate the gene we want. Then in comes DNA ligase, which acts as a kind of molecular glue, seamlessly pasting that gene into a new piece of DNA. This simple, elegant cut-and-paste system, it's the foundation of all gene cloning.
Okay, so our gene is cut out and ready to go. How do we deliver it? Well, it turns out instead of reinventing the wheel, scientists often look to nature's own experts in gene delivery, viruses and bacteria.
These natural carriers, which we call vectors, have had millions of years to perfect the art of injecting their own genes into cells. And this is where scientists got really clever. They figured out they could essentially hijack these delivery systems. They can remove the virus's harmful genes and replace them with the one they want to add. Then, the virus just does all the hard work for us.
A fantastic example of this is the cauliflower mosaic virus. This little plant virus is incredibly easy to work with. Get this, you can literally just rub it onto a leaf and it starts infecting the cells, making up to 1 million copies of its DNA and our new gene inside every single one. It's like an unbelievably efficient natural delivery truck.
But what happens when a natural vector just won't work for the organism you're studying? Well, sometimes you've got to be a bit more direct. This next batch of methods basically bypasses biology altogether and uses pure physics to punch through the cell's defenses.
Picture a cell's outer membrane as a locked door. Electroporation is kind of like giving that door a quick, high-voltage zap. That jolt forces tiny, temporary holes to open up in the membrane, just long enough for the DNA that's floating outside to slip right on in before the door seals back up.
And this method is shockingly powerful. For something like E. coli bacteria, you can get what's called transformation efficiencies up in the billions. That's up to 1 billion successful gene transfers. It's fast, it's simple, and it's a super effective way to modify a massive number of cells all at once.
Now, if zapping a cell doesn't do the trick, how about just shooting the DNA directly into it? It sounds wild, but that is the basic idea behind particle bombardment, which uses a device that is, I kid you not, literally called a gene gun.
The whole process is basically like loading a microscopic shotgun. You take these tiny heavy particles, usually gold or tungsten, and you coat them with the DNA you want to transfer. Then you load these DNA-coated bullets and use a blast of gas to fire them at your target cells. The particles are just small enough to pierce right through the cell wall and membrane, delivering that DNA cargo right inside. This brute force method was a total game changer for modifying plants like corn, which have really tough cell walls that were a huge barrier for other techniques.
So why use a precious metal like gold? It's not just to be fancy. Gold particles can be made with a very uniform size and shape, which is absolutely critical for getting consistent results. But even more importantly, gold is chemically inert, which means it's way less toxic to the cells it's being blasted into.
Okay, so from a shotgun approach, we now move to the sniper rifle. This is the most precise, almost surgical method of them all. Microinjection is exactly what it sounds like.
You use an impossibly tiny glass needle to inject a DNA solution directly into one specific cell, often right into the nucleus. This is the go-to technique for things like creating transgenic animals, where that level of precision is everything.
So with this method, you get unbelievable precision. You know for a fact which cell got the DNA. It can punch through cell walls, and it works on almost any organism. But all that precision comes with a hefty price tag. It's a very slow, very expensive process that takes highly skilled people to pull off. One tiny wrong move, and the cell is toast.
So, we've covered hacking nature, and we've seen brute force. But there is another way, and this one is all about finesse. It's about using chemicals to gently persuade a cell to take up that new DNA.
One of the most common ways to do this is with a chemical called polyethylene glycol, or just PEG. You can think of PEG as a sort of chemical doorman. When you mix it in with your cells and DNA, it kind of destabilizes the cell membrane, making it more fluid and open. This coaxes the cell to just absorb the DNA floating around it in the solution.
The huge advantage here is control. You're not relying on a virus. You know exactly what DNA you're putting in. The major downside, though, especially in the plant world, is that this only really works well on what are called protoplasts, cells that have had their tough outer wall chemically stripped away. And then trying to regenerate a whole healthy plant from one of those fragile wall-less cells, that's a massive technical challenge all on its own.
So we've got biological vectors, we've got physical force, we've got chemical persuasion. With all these options on the table, how does a scientist actually decide which tool to pull out of the toolbox? Well, it all comes down to a series of trade-offs. So what this all really tells us is that there is no single best method. It just doesn't exist. Biological vectors, super efficient, but you give up some control. Physical methods, you get pinpoint accuracy, but you risk damaging the cell. And chemical methods give you great control, but they come with all sorts of other technical hurdles. The right choice always depends on what you're working with and what you're trying to achieve. All right, let's just pull back for a second and look at the big picture here. Why does any of this matter? Well, because mastering these techniques is the key that unlocks the door to pretty much all of modern biotechnology. Seriously, every time you hear about bacteria being engineered to produce insulin for diabetics, or corn that's resistant to pests, or a groundbreaking new gene therapy, it all starts with one of these fundamental methods. This isn't just a random collection of lab tricks, it's the absolute essential toolbox for building the future of medicine, agriculture, and so much more.
From hacking viruses to firing golden bullets, we've come up with an absolutely astonishing set of tools to edit the very blueprint of life. The question isn't just about what we can do today. As these tools get even more precise and even more powerful, you have to ask, what incredible new possibilities will we choose to unlock or rewrite next?